Folate's role in managing chronic fatigue is a quiet but compelling example of how a single micronutrient can influence energy at its biochemical roots. While fatigue is often dismissed as a vague, nonspecific complaint for many individuals, especially those struggling with chronic fatigue syndrome, CFS, or persistent low energy, this symptom reflects deep physiological imbalances that are rarely addressed with surface-level treatments. Folate, or vitamin B9, is a critical component in multiple pathways that generate cellular energy, maintain mitochondrial function, and support neurotransmitter balance. When folate status is suboptimal, these systems falter, leading to a kind of invisible exhaustion that is not psychological, but biochemical. At the core of this fatigue is folate's essential contribution to the 1-carbon metabolism pathway, a complex network that feeds directly into mitochondrial energy production. This pathway involves the transfer of single carbon units for the synthesis of nucleotides, amino acids, and methyl donors, all vital for proper cellular metabolism. In particular, folate is required for the generation of adenosine, a precursor to ADP, the body's primary energy currency. Without enough folate, ADP production can slow down at the mitochondrial level, depriving cells of the energy they need to function optimally. This is not limited to muscle cells or neurons. It affects every tissue, contributing to a global sense of fatigue that no amount of sleep seems to resolve. Mitochondrial dysfunction has emerged as a central theory in the pathophysiology of chronic fatigue syndrome, and here too folate offers potential support. Mitochondria rely not only on nutrients like CoQ10 or carnitine, but also on methylation to maintain their DNA, optimize protein synthesis, and regulate oxidative stress. Folate, by donating methyl groups and facilitating same acetylmethionine production, enables the repair of mitochondrial DNA and the modulation of gene expression that governs mitochondrial efficiency. In folate-deficient states, mitochondrial enzymes may become less effective, oxidative damage may accumulate, and the entire organelle can slip into a low-functioning mode. Supporting folate status in this context is not about simply giving a vitamin. It is about restoring mitochondrial competence at the epigenetic level. Another mechanism by which folate helps combat chronic fatigue is through its role in neurotransmitter synthesis. Low energy is often accompanied by low mood, brain fog, and lack of motivation, symptoms strongly tied to imbalances in serotonin, dopamine, and norepinephrine. These neurotransmitters are synthesized from amino acids in methylation-dependent reactions that require folate. When methylation is impaired, the brain's ability to maintain adequate levels of these chemicals diminishes, leaving individuals not only physically tired, but also mentally depleted. In many people with chronic fatigue or subclinical depression, addressing folate deficiency, especially when combined with vitamin B12, has been shown to improve motivation, concentration, and emotional resilience, indirectly lifting the burden of fatigue from multiple angles. Interestingly, folate also helps reduce levels of homocysteine, a byproduct of methylation that, when elevated, can interfere with energy production by impairing endothelial function and reducing blood flow to tissues. This vascular effect is subtle but crucial. Efficient blood flow ensures that oxygen and nutrients reach mitochondria efficiently, and when circulation is compromised, tissues experience an energy bottleneck that may contribute to the pervasive fatigue reported by individuals with chronic inflammation or cardiovascular dysfunction. By keeping homocysteine in check, folate preserves this vascular tone and indirectly enhances energy availability at the tissue level. Genetic factors also play a role in folate's impact on fatigue. Individuals with MTHFR polymorphisms, particularly the C677T variant, have a reduced capacity to convert folic acid into active 5-methyltetrahydrofolate, which is the bioavailable form used in methylation. For these individuals, even a diet adequate in folic acid may not be enough to restore full metabolic function. Supplementation with L-methylfolate can bypass this bottleneck and improve outcomes in those with unexplained fatigue or poor response to standard interventions. Finally, folate's role in immune modulation adds another layer to its fatigue-fighting potential. Chronic fatigue is often associated with low-grade inflammation or immune dysregulation. Folate participates in the regulation of T-cell proliferation and cytokine expression through its influence on DNA synthesis and methylation. By maintaining immune balance, folate helps prevent the chronic immune activation that often underlies fatigue, malaise, and non-restorative sleep. In all these ways, supporting ADP production, optimizing mitochondrial function, balancing neurotransmitters, enhancing blood flow, and modulating immunity. Folate functions as a quiet yet foundational force in energy metabolism. For those grappling with persistent, unexplained fatigue, it may be the missing link between dysfunction and vitality. And though it often operates silently, when folate is restored to optimal levels, the result can feel like a reawakening from a long, biochemical sleep, a return of energy, clarity, and the drive to fully engage with life.